without further ado, we've got Carol Stoker. Carol was one of the key founders of the Mars Underground way back in 1981, where a group of then graduate students at UC Boulder decided they would put the objective of humans to Mars back on the programmatic map for NASA, which had retreated into thinking that its purpose was to launch weather and reconnaissance satellites and leave these things to disregard them as the dreams of youth, and which I'd always been interested in Mars, but which ultimately, though, recruited me to actively devoting my life to making humans to Mars a reality. I was not at the first Case for Mars conference. I was at the third, and after that. But and then, of course, after graduating, she went to NASA Ames, where she's been a mover and shaker in all sorts of initiatives, some realized, many not, to advance the cause of the exploration of Mars, in particular the search for life, drilling on Mars, and other projects. So without further ado, Carol Stoker.
Okay, well, I hope I can make these fancy devices here work. Okay, it did work. It works not as well as it's supposed to. Okay. All right, so I'm going to talk to you about what is the kind of the current things that are going on the search for life in the solar system. And it's kind of boiling down to, in my view anyway, two very interesting targets, Mars and the geysers, literally geysers, that are happening on the moon of Saturn, Enceladus. Okay, so where should we look for life? This is basically what this arrow is pointing to is the increasing chance of life being not related to Earth. So basically, starting with Mars, it's our nearest neighbor. It's the most habitable terrestrial-like planet. And it had certainly the possibility of life early in its history. It had liquid water. It had a thick atmosphere. However, its current surface is cold and dry. There could be refuges for life possible in the subsurface and the near-surface ice, but in general, it's not a very attractive planet. We've spent a lot of time on it, and clearly there are not giraffes walking around on it. So the next target that's under consideration is Europa, which is, of course, a moon of Jupiter. Appears to have a subsurface ocean beneath a very thick ice cover, and there's an argument that that ocean could be habitable. And then finally Enceladus, which I mentioned, apparently has subsurface water, again, beneath the thick ice cover, but it has geysers that are shooting that out into space. So we actually have no idea how life got started. There have been sort of 4 models proposed, that panspermia being one of them, that life came to Earth from the stars via comets, that life may have come to Earth from Mars because early in their history they were very similar and they actually exchanged material. Probably the dominant model is that life developed in sort of the warm, wet pond, which actually required there to be land surface area on the Earth. And it was probably hot. It was hot water. And then a 4th model is that it actually came from sea, again, deep ocean vents of hot water. And so that origin of life can be tested by searching for life if we actually find other examples of life elsewhere in the solar system. So for example, for Mars, Earth is, the only example of life we have. For Mars, if we find that same example of life, that's going to argue for Earth-Mars exchange of life. There's really no case for there to have been exchange of life between Earth and the icy moon. So that if we find life there, it's almost certain to be a different example of life. The cometary source, they could all be the same example. If life began on land, we would expect that life on Earth and Mars might be different, and there should be none on the outer icy moons of the outer solar system. And then finally, the deep sea vent, there could be 3 different examples of life. And so by different, what I really mean is biochemically different. And life is currently, we have only one example, so we only know one way for life's biochemistry to behave. So the only mission that has ever actually searched for life was the Viking Lander in 1976. And you can remember how long ago that was, because this is Carl Sagan standing in front of a model of the Viking Lander. So there were 2 landers. One was landed at mid-latitudes and the other at high latitudes. There were, on the lander, there were 3 life detection instruments, very sophisticated little package. A lot of it was built at Ames Research Center, as a matter of fact. And there was an organic analysis instrument, which is similar to the instrument that's currently on MSL, the SAM, or Sample Analysis on Mars. There was also a robotic arm that scooped soil into the instruments. So what were the Viking biology experiments like? Basically, these were, again, they were very sophisticated for the time that they were invented. They were attempted to culture soil microorganisms, because microbiology at the time, that's really all that was known how to do, was grow up what it is that's living there. And so they basically exposed the soil to a nutrient broth, and they looked for exhaled gases as evidence of metabolism. Again, there were 3 of them. 2 of them actually behaved identically, or not identically, but with a reduced response when they were, after the soil had been heated, sort of a sterilization. The 3rd one actually was totally consistent with what you'd expect for life, producing the response. And in fact, the PI of that experiment, I'm surprised he's not here. He still to this day insists that he found life on Mars. So the results were that the soil was unexpectedly reactive. The reactions behaved somewhat like Earth life would produce, but they were different in detail. But the organic analyzer showed no organics in the soil, 0, 2 levels of parts per billion. And this was argued to rule out a biological explanation. So, as anyone knows who watched Star Trek, there's a great little instrument in the Star Trek era called a tricorder. And with the tricorder, you can just point at something and know it's alive. It's life, Jim, as Spock said in almost every episode. So, is there a tricorder? The answer is no. How do we recognize life? We're actually, since Viking, there's basically been no money going into inventing a tricorder or trying to make an instrument like this. So it's only just recently that this has become a topic of interest that has started to draw some cash in, and there has been various workshops and trying to understand how we would recognize life and what we should do. So basically, the search for life, as we would argue, is actually dead. It's not, you should not be searching for something alive. I think as we learned from Viking, you should not try to do a metabolism experiment. We actually know now that most things from extreme environments cannot be grown in culture. We didn't know that at the time of the Viking experiments. At the scale of microorganisms, shape is not convincing. So the best evidence of life is actually dead, and it is biomolecular structures. And in particular, it's complex biomolecular structures. So it can be patterns of molecular weight, and in one aspect is its chirality. It is the handedness of amino acids that make up life, because life has to have a handedness bias. Amino acids from space, from meteoritic amino acids are kind of balanced in terms of left and right-handed molecules, but life can only pick one-handedness in order to make the more complex biomolecules. It could be either hand. On Earth, it happens to be left hand. But so looking for a chirality or bias, a bias in chirality, is one of the ways that people now think one should go about finding evidence of life. So what has NASA been doing? Here's an example of what NASA's been doing a lot of since Viking. This is a Curiosity rover. It's really good at taking its own picture. This is a selfie. Landed on August 5th, 2012. As a matter of fact, there was a Mars Society convention when it landed in Pasadena. I was there. It was exciting. In the 5 years since it landed, it's traveled 17 plus kilometers. Its instruments are focused on geology and mineralogy, and accessing habitability, which is, let's go look for evidence of water. And it's found plenty of evidence of water. There was definitely liquid water at this site, no question. But what has it found that's relevant for life? So I can only pick out 3 important findings that are relevant for searching for life. One of them is that you need to get below the surface. These are the different drill holes that have come from the long traverse. The original idea of this landing is it was going to land here and go straight up, climb Mount Sharp, which is actually in this direction. But it turns out there's this huge sand deposit in the way, which they had to drive around. So they've been driving along this thing for the whole 5 years. But they've drilled in various spots, and they have found that red Mars is only right at the surface. And when you get under the surface, you find gray Mars. And that's important because that means you have reduced compounds, that it is not oxidized. And furthermore, they have found that the darker gray materials actually do contain organics. And there's a variation of how much organics you find depending on where you drill. So, but then there's this other issue about organics, and that comes up with this, this next thing. And this is really interesting. It turns out in 1964 there was this movie, Robinson Crusoe on Mars. and in this movie, there were these yellow stones which burned like coal and released oxygen. And that was kind of one of the themes of the movie. That's how he stayed alive. Well, turns out there are yellow stones on Mars that burn like coal. Who would have thought? 1% of the surface of Mars is this mineral perchlorate. It's a salt, actually. Its chemical formula is magnesium or calcium and a chlorine atom and 4 oxygen atoms. Why is it important? Well, from the point of view of understanding what happened with the Viking mission and why it didn't see organics, is that it burns organic carbon. In fact, it burned the organic carbon from the soil put in the Viking GCMS, because the way a gas chromatograph mass spectrometer works is you have to heat the sample up to release gases, and those gases get sucked into the mass spectrometer for analysis. Well, that heating process actually allows the oxygen to get released, and the chlorine and oxygen be very strong oxidants, and they produce carbon dioxide and chlorofluorocarbons, which on Viking were interpreted to be cleaning fluid, remnants of cleaning fluid. It was actually on SAM they recognized that this probably wasn't cleaning fluid and recognized that it was a signature of organics. But they don't see the spectrum of organics they should see because they're being oxidized. It's food. Many microbes know how to metabolize, use perchlorate to metabolize reduced substances. And it's antifreeze. It actually can lower the freezing point of water to typical temperatures in Mars polar regions today. Now, you can't just take perchlorate and stick it on ice and make a habitable solution because basically you get a eutectic and the water activity at that eutectic is not habitable, at least for terrestrial organisms. But it's actually an explanation for a lot of things that are going on Mars, like the recurring slope linea, the fact that at night the soil seems to suck up water, which it releases during the day. It seems to breathe water. So there's a lot that can be explained by perchlorate. So the lesson is you need to characterize the chemistry. And I'm way behind schedule. No. Okay, so another lesson is follow the biosignature. So, so basically, SAM detected methane, variable at 5 to 10 parts per billion. It was detected on 4 separate occasions over a 60-sol period. Well, methane is a biosignature. It's produced by microbes. Without biology, it can be produced, but it requires a water rock reaction with a very unusual mineral called serpentine, which means there has to be liquid water and serpentine interacting, and episodically. Its presence is unexpected and hard to explain. So what have we learned about life on Mars so far? I was going to leave this blank because we've learned so little. We know that early Mars was habitable. We know that conditions on current Mars are at or below the limits of what terrestrial biology can survive. And we've not found any evidence of life, but we haven't actually looked. We can't recognize fossil evidence because we don't have the right instruments, and we can't recognize organic evidence because the instruments we've flown have been foiled by perchlorate. So the next mission plans to basically go collect samples and cache them to bring back to Earth to look for evidence of ancient life, not modern life. There's a couple of problems with this mission, which I won't go into since I'm running behind schedule. But what are they going to look for? Well, basically what early life on Mars looked like was stromatolites. These are coral-like, things like coral heads. And they are the earliest evidence of life on Mars, and they actually have been identified in these deposits in Western Australia, 3.5 billion years old. And when you take these rocks, and by the way, there are modern examples of these, also in Western Australia, and you can actually see both in one day. You can go to the most ancient evidence of life and a modern analog in the same day. Take them and cut them in cross-section, and they are complex layered structures. In very rare cases, they might host fossil bacteria. But proving that bacterial fossils are real evidence of life is really difficult. You need microscopic imaging of the cellular structure. You actually, to be convincing, you need to be able to see the cellular structure divide. They have to have preserved organic carbon in them at the microscale. And you have to have a modern analog that is identical for comparison. Can we do that on Mars? I think it's unlikely. I've been an advocate of a mission called Icebreaker, which has been proposed to Discovery last in 2015. We'll probably try again. But this mission goes to the polar regions where there's modern habitable environment, not contemporary, but in the recent past, and drills a meter into soils that should have thawed. So there should have been liquid water in those soils less than 1 million years ago. Okay, so recently Congress changed the game Some people thought it would take an act of Congress to get NASA to do life detection again after Viking. And in 2016, Congress complied and directed NASA to create an ocean worlds exploration program whose goal was to discover extant life on another planet. So in response to that, NASA has announced a lander mission to search for life on Europa, which is supposed to land in 2026, Europa being one of the 4 moons of Jupiter. Europa may have a subsurface ocean. The 2 lines of evidence for that are that it distorts the Jovian magnetic field, suggesting that it's conductive, and its surface is fresh, there's no craters on it, and it looks like fractured sea ice. So the idea is that there are cracks forming in this surface ice shell, allowing fluids to come to the surface. That ice shell, however, is hundreds of kilometers thick. So again, the model for this is deep sea hydrothermal vents, and this is the type of life that could exist on Europa. But the surface of Europa is a very hostile environment. It's embedded in charged particles trapped in Jupiter's magnetic field lines, the so-called Van Allen belts. I don't think they're called that on Jupiter, but the radiation dose is 540 rems a day. And so organics on the surface will be very quickly destroyed by radiation. Because the lander has to endure this high radiation, the mission will last only 30 days. And you can see that what they're planning for is a tiny little lander. It's 1 meter square. And because of the high radiation, which is also going to destroy organic molecules very quickly, the samples have to come from at least 10 centimeters depth, so they have to have some kind of a drilling-like device. They're not planning for an actual drill, but it's a difficult mission to achieve. But there's this other target, Enceladus, and it's a very tempting target for life. It's only 500 meters across, but it's the brightest object in the solar system because geysers are spewing gas and ice out of its subsurface, and actually is forming one of the Saturn rings, the E ring. And its surface, like that of Europa, is fractured. But the Cassini mission has imaged the fact that there are plumes coming out. It's a very specific area. It's called the Tiger Stripes. They're in the South Polar region, and they're shooting ice out at 400 meters per second. It extends hundreds of kilometers out into space. All you need to do is fly by and grab it. So Cassini, next Friday, is going to dive into the Saturn, is going to end its mission by diving into the atmosphere of Saturn. Studied Saturn and its rings and its satellites for 13 years with 15 science instruments. It's one of the most successful missions of all time, I have to say. And one of its really stunning results was that an instrument, an ion and neutral mass spectrometer, which was really not designed to do this at all, but captured gases from these plumes, and lo and behold, there are biosignatures, potential biosignatures, in those gases. There's methane, simple organics, relatively complex organics. Again, the instrument wasn't designed to measure biomarkers by any means, and recently it's been reported in Nature they actually see free hydrogen. So there's everything. If you were to just dump microbes in there, there's everything that they would need to live off of. It doesn't necessarily mean the origin of life can occur in that environment, but there's definitely a habitable environment. And all of these organics could be biosignatures. So the most recent thing that's happened is that, again, Ames Research Center, with Chris McKay as PI, and a combination of Goddard and APL, has proposed 2 New Frontiers program, a mission called ELSA, named for the Disney character that's in the Ice World, or Ice Queen. The idea here is that you get a spacecraft to fly through these plumes with a collector that sweeps up ice particles and analyzes them for biomolecules. The mission is in review. Actually, it's been reviewed. It's in final decision. We should know really within the next few days, few months, whether or not it gets selected for further study. It isn't selected for flight, even if it does win that initial competition. But, so that's what's going on. And my time's up. Jim.
Yeah, several years ago there was a bit of a furor about some structures that were found in the Mars meteorites that were picked up in Antarctica, and I never did hear that there was any real resolution to whether those were indicative of life or not. Could you comment on that?
Yeah, so what you're referring to is, it was proposed by Dave McKay and his colleagues, who were actually meteoriticists. They studied meteorites. And they found and announced in the mid-1980s evidence, or late 1980s, what they purported to be evidence of biology in a particular meteorite. I actually had that in the talk, and I cut it out because I had too much. I thought I didn't have enough time, but they purported evidence of life from this essentially fossil bacteria. And this is, to me, this is a lesson on how hard it is to prove that fossils are really evidence of life. This discovery actually started the entire astrobiology program, but kind of the first thing that happened is, and this is often what happens, this is actually an example of the history of trying to identify the earliest evidence of life on Earth, is that a huge number of scientists requested and got samples of this same meteorite and looked at it and drew their own conclusions, most of which were focused on arguing with the case that the McKay et al. Team had made. To my mind, their evidence was pretty good. They had 3 lines of evidence. They had organic carbon, they had cellular structures, and they had a 3rd line, which to my mind has still never really been refuted, and that is that they had chains of magnetite that were associated with these cellular structures. It was the chains of magnetite that I thought was the most compelling part of their argument, because it's actually very difficult to put magnetite into chains like that. But the community, as a consensus, in my view, kind of ganged up on them and refuted their claims. And typically the refutations were they would knock out one or the other of the 3 claims. They never really knocked out the magnetite part. But the consensus at present is that that meteorite does not have biosignatures. But I think it's still kind of an open question.
My question relates to the depth to which one needs to drill to find evidence of life if it should exist. I assume that the protection from soil or ice is proportional to the thickness, and there's not an absolute that 99 centimeters everything dies and 101 everything survives. Since we don't know what we're looking for and how radiation resistant it is, what's the risk that we could miss something if we drill 10 centimeters or 100 and we have a less radiation resistant organism that might survive at 2 or 3 meters deep but not at 1 meter?
So I'm not sure if you're referring specifically to the sorry, drank that the wrong way. Down the Enceladus, the Europa case. Right, and that's the Icebreaker plan. So if anything sits on the planetary surface long enough, just natural radiation will break down its DNA and won't be able to reproduce. So there has to be periods where reproduction is possible. So the Icebreaker, and there's a paper on this, you can look it up, you can read the logic, but basically at 1 meter with episodic warming, which does occur because Mars goes through these climate change cycles that are quite dramatic compared to anything the Earth goes through. There is a argument to be made that drilling 1 meter, you will get to materials that have had habitable conditions within a time scale that they could also have survived radiation damage, because they can reproduce periodically. So they have to be able to wake up and do reproduction on time scales of something like 1 million years in the upper meter. So if they were deeper meaning below, say, 2 meters, then they would not be destroyed by radiation damage, at least cosmic ray radiation damage on those kind of time scales. But they also don't, at least for that model, the thermal pulse doesn't get down that deep. So any life on Mars that isn't living in sort of that upper couple of meters actually has to be very deep, like deep enough for there to be liquid water from the, basically the thermal heat coming out of the interior. And that might be kilometers.
Yeah, it has to have some energy source. I've seen some pictures that indicate that there have been very recent, in fact, in the last few years or less, water flows. Could this antifreeze stuff? I've heard some people say, well, it's salts. And yeah, you could get salts, but salts don't have a good way to get uphill to allow more flow. But I think that this antifreeze you're talking about, or this antifreeze-like substance, could conceivably do that.
Actually, what you're referring to is the recurring slope linear, and one of the explanations that's been offered for what is allowing that flow, because clearly there's something happening that looks like flow, is that the other thing that I didn't mention about perchlorate is it's extremely hygroscopic. So if there's any water vapor around, it sucks it in and basically turns its crystals into drops. And so if you get enough of that happening, you could get flow. However, more recent papers have been looking at the actual angle that flow is coming from, and it turns out that it looks, from a point of view of the frictional properties, that it is dry flow. So I think we don't really understand the recurring slope linear. They definitely look like aqueous flows if you just see a movie of them. But there's something more complicated going on. It looks like we actually have to stop.
Yeah, sorry, time constraints. We have to move on. So.